Welcome to Analytically Delicious, Insights into Food Safety by LVA. Join us as we explore the latest developments, trends, and challenges in ensuring the safety of our food supply through analytical techniques, uncovering the science behind what makes our food safe and delicious.
Welcome to Analytically Delicious. I'm David Steiner, your host, and today we dive deeper into the area of food safety and risk assessment through the lens of analytical chemistry. In our ongoing exploration of critical scientific issues, we have examined the complexities of residue and contaminant analysis, highlighting the essential role of robust analytical methodologies in ensuring the safety and quality of our food supply. Joining us is Michael Sulyok, an esteemed analytical chemist and leader of the Multi-Toxin Analytics group at IFA Tulln. Michael's groundbreaking work in developing comprehensive multi-method approaches for mycotoxin analysis has significantly advanced global standards in food safety. Analytical chemistry and multi-method approaches are crucial in food safety as they provide precise tools to detect and quantify contaminants like mycotoxins across various food matrices. By enabling thorough analysis and identification of potential risks, these methodologies empower regulatory bodies and food producers to implement targeted interventions, ensuring consumer health and confidence in the food we consume. Michael will share his insights into the challenges, innovations, and future directions of analytical chemistry in safeguarding our food supply. Michael, in our recent episodes, we explored the world of mycotoxin and pesticide analysis and concluded with a session on food safety with Rudolf Kriska. One key takeaway from this session was that proper risk assessment requires a comprehensive and also robust analytical data foundation. And this is where you come in, as you are a globally recognized analytical expert, renowned for your achievements in developing the most extensive LC-MS-MS-based quantitative multi-toxin method. Michael, I'm really curious now, how many analytes can you typically detect in a single run?
Well, the current method version comprises 1,000 natural metabolites, 900 of them being mycotoxins/fungal metabolites. And we have also implemented some major plant toxins and a few bacterial toxins. Mm-hmm. But at the moment, we are working on a further method extension, and it will be something like 1,250 compounds afterwards.
That sounds really impressive. And what are the typical application areas for your comprehensive multi-toxin method? Or can we actually say multi-class or multi-compound method?
Yeah, well, since the routine method we are applying is combines plant toxins and mycotoxins and a few bacterial, as I said. But we have an add-on module on demand for pesticides and veterinary drugs as well. Mm-hmm. And when it comes to application, well, we have both academic cooperations and more like service type of cooperation. Mm-hmm. So part of them is like fungal metabolite profiling, so where our cooperation partner wants to know what can a particular fungal species produce on optimized media. Then it's like more like the influence of different conditions like growing conditions and different stimulants on the fungi, on the overall metabolite profile. And of course, when it comes to food/feed safety, we receive different kinds of samples where our collaboration partner is interested not only on the mycotoxins addressed by regulatory limit, but on all the other fungal metabolites for which there is no toxicity data, but potentially they might be harmful. Mm-hmm.
So it's a quite broad application area, we can say. And can you also share a best practice example from your career where insights gained through your extensive multi-toxin method led to discoveries and also conclusions that would not have been possible otherwise?
There was some 10 years ago, we were approached by a veterinarian from South Africa, and they had a flock of cattle showing serious tremors, and I think, if I remember correctly, also some animals died. Mm-hmm. And they traced it back to moldy sprouted barley, so that was actually a technique coming from Canada, so you grow this barley and still and harvest sprouts. But the problem was in South Africa that actually the temperatures were too warm, and so what they observed was black spots on this moldy barley, and they thought, well, this looks like aspergillus flavors, and so they sent the sample to a contract lab. Because they thought, okay, well, it's aflatoxins, which doesn't fit to Dramagens, but still this was their first sort of explanation. And that contract lab, which analyzed the usual mycotoxins addressed by regulatory limits, told them, well, the samples do not exhibit something special. And so they sent the samples to us, and we analyzed them with our method. And what we actually found, it was indeed Aspergillus, but it was not an aflatoxigenic Aspergillus, but Aspergillus clavatus. And this species is able to produce patulin and cytochalasine E and some triptocubalines, which are all sort of known as biologically active substances in vitro experiments, but actually no one else analyzed them, maybe except patulin in fruit-based product. And the levels of patulin were some 50 times higher than the current regulation that is set for food. That's still no full proof evidence, but it was actually sort of a good indication that this particular Aspergillus caused a problem.
I understand. You said that was about 10 years ago. That means that you had already a quite broad method in place. However, I would be interested how has these multi-methods in general developed and improved over the years and decades?
Well, I joined the IFA Tulln back in 2005, and at that moment the new instrument was acquired, and sort of this was actually the beginning of some scientists giving presentation on multi-mycotoxin method, but at that time it was like trying to accommodate all the regulated ones. And sort of my first small project as a sort of young postdoc there was to try to accommodate some 40 metabolites in one single method, and sort of the expectation where, okay, we analyze diluted crude extracts, and probably this will not be quantitative, but should be okay for a semi-quantitative screening. But sort of the outcome of the study was, okay, there are some effects, some matrix effects deriving from the constituents of maize, but if you keep them under control, you can really do quantitative analysis.
Matrix effects, can you maybe briefly explain this term to our listeners?
Well, in LCMS, matrix effects denote the influence of the matrix on the ionization of analytes. So typically, what is observed that if you spike a blank extract with a known concentration of your analyte, then the analytical response is different to a solvent-based standard of the same concentration. And sort of a very crude explanation is that actually co-eluting compounds from the matrix, so fats or carbohydrates or proteins, compete with the analyte in the ion source of the mass spectrometer for the available electrical charge, and thus influencing signal intensity.
Understand. So that means that matrix effects arise from the compositional characteristics, as you said, coming from the macronutrients like carbohydrates, fats, and so on, from the test material, which significantly impacts the analytical outcome. Are these matrix effects consistent within a matrix commodity group, or do you observe any variations?
Well, we published some 4 years ago a paper where we investigated different types of grains, dried fruits and nuts, and what we saw is that the matrix effect within a commodity group are quite diverse. So I would not really sort of agree with the statement pointed out in some regulations that you need to validate just for one type of commodity for each commodity group. So I think the matrix effects are significantly different. But even within a given type of matrix, let's say maize, we found that there are variation in those matrix effects between maize variety, let's say coming from Austria compared to like white maize from Africa lacking the beta carotene. And actually our conclusion we've drawn in our recent papers was that actually characterization of these relative matrix effects, meaning that how big is the variation in the absolute extent of matrix effects between different individual sample within a given matrix, is very crucial to come up with a realistic estimation for the expanded measurement uncertainty.
Mm-hmm. So you have already mentioned the regulatory aspect. How are matrix effects addressed in the guidelines and in standards? Do you think this issue is given sufficient attention, or is it somewhat neglected in the current regulatory practices?
Well, I think that the absolute matrix effects are sufficiently covered, but those relative matrix effect I was just referring to, I think it's only the very recent regulations on mycotoxins and on veterinary drug residues that they have specific points refer to this issue. So they recommend either to investigate the matrix effect in 20 different individual samples, or in case of the mycotoxins, plant toxins, they strongly suggest to support validation data deriving from technical replicates of the same sample with ongoing results from proficiency testing.
So I think it's very clear that within an analytical lab you want to get rid of these matrix effects. What are the typical compensation and mitigation strategies which can be employed to address matrix effects in analytical testing?
Well, there are a few options. The most straightforward one is to further dilute your sample, so it's like diluting out the matrix, but that runs into the obvious issue of the sensitivity of your method being compromised. So there were some studies on pesticides conducted which have shown that if you really want to completely dilute out your matrix, you might need to dilute the extract up to 1,000-fold, which is of course not feasible if you need to obtain LOQs in the low microgram per kilogram range. The next option would be some unspecific cleanup methods like catchers, which was actually designed some already 20 years ago for pesticide residue analysis. So the idea there is that you have a rather crude phase separation step where the idea is that you get rid of the dirt in the lower water phase. We gave this a try, but it was not very convincing for our method. So there was not a big improvement for most of the analytes, where there are a few very polar compounds that actually partition into the water phase and are lost. So that's why we do not apply catchers. And the last option is the use of stable isotope label internal standard, which are commercially available. And I think this is the way to go for the official control labs, which focus on the mycotoxins addressed by regulatory limits, because for all those compounds, the related internal standards are available. And this approach has to be shown to, really compensate both absolute and relative matrix effects in a very convincing way. So if I would be just an official control lab looking at a limited set of samples, I would choose this option, no question.
However, the most straightforward solution is the dilution step, as you mentioned, and this means that if your instrument provides sufficient sensitivity, these effects can be compensated by a simple dilution step applicable to all compound groups, right? However, are there any other technical limitations beside sensitivity that need to be considered?
Well, of course, the obvious drawback of liquid chromatography coupled to tandem mass spectrometry is that data acquisition is sequential, meaning that for a given time period, typically in the range of 20 milliseconds, the instrument just acquires data for one given MS-MS transition of one analyte. And so if you want to accommodate several hundred of compounds with 2 MS-MS transition per compound, actually the sort of the speed of the mass spectrometer very soon becomes the limiting factor. So what is usually done is that you define for each analyte the expected retention time plus minus a certain window width for which this compound is scanned. But still, it's the time span for acquiring one data point, the so-called dwell time, is the limiting factor. And if it becomes low, it severely affects the analytical quality of the data, mostly the reproducibility.
So do you typically run this method under HPLC or UHPLC conditions? And is this dwell time you mentioned also affected by different chromatographic solutions?
Yeah, sure. I mean, UPLC has the obvious advantage of narrower peak widths and increased signal intensity, but at the same time, it would actually make the problem of dwell times even more pressing one, because if the peak gets narrower, you need to acquire the required data points per peak in a shorter period. And in principle, you could argue that if the peaks get narrower, you could also narrow the time window in which you acquire one given analyte. But still, this requires the retention time to be super stable. And we gave UPLC some tries, but we saw that actually the approach we use to run a very large column with 4.6 inner diameter with a very high flow rate actually provides super stable retention times. And the other aspect is that we are running this method with a very crude sample preparation. So we use just an acidified acetylnitrile water extraction, followed by a dilution step, so the sample extracts are sometimes turbid, so contain a lot of matrix. And with our very large column, this does not pose any problem as considers the stability of chromatography. So we are running a column usually for years. And to put it sort of in another way, is like for such analysis with a very crude extracts, the robustness of a HPLC column is probably the more important aspect compared to the better analytical performance that a UPLC column would provide in terms of sensitivity. Or to put it in other words, if you want to compete in the Rally Paris-Dakar, you are not going to choose a Formula One car as your vehicle.
Absolutely right, yeah. Michael, a different question. Since we have discussed in all our previous episodes how high-resolution mass spectrometry can potentially replace triple quadrupole technology, what you are actually applying, why have you not yet implemented a high-resolution instrument in your routine workflow?
Well, high-resolution mass spectrometry in scanning modes has for sure the big advantage of the possibility of retrospective analysis, which means that years after you've acquired a data set, you could check for a new compound popping up. So that's not feasible with LC-MS. But high-resolution mass spectrometry has also some, I would say, disadvantages. One which I would refer to maybe until a few years ago is a decreased sensitivity compared to the triple quadruple mass spectrometry. From what I get from talks from colleagues at conferences, probably this gap has now mostly been closed. But I wonder with high-resolution mass spectrometry whether you might also run into the same problem like for MSMS, which considers the scanning time. Because if you go to regulations, they require that you actually acquire one product ion in high-resolution mode. And I wonder if you accommodate, like we do, several hundred analytes, and you potentially need to acquire product lines for all of them if you do not run into the same issue of limited scanning time. And that's actually something we will for sure investigate in the future.
I understand. And considering all the advantages you highlighted by applying your LC-MS-MS-based multi-method, how do these advantages translate into practical benefits in everyday laboratory operations?
Well, the obvious advantage is that you have sort of one workflow with one sample preparation protocol. Yeah? If you need to apply methods which are suggested by European normative normalization and want to test for compliance for, let's say, I don't know, all regulated mycotoxins in maize, you would end up maybe with 6 different methods where you have to aliquot your sample and do 6 different sample prep protocols, which is, of course, an enormous effort in terms of personal costs and material costs. And yeah, the obvious advantage is to reduce workload and work costs.
And last question, Michael, is there any drawback associated with such an approach that prevents a routine laboratory from implementing it?
Well, maybe it's not a drawback, but rather a consequence. If you're acquiring a large comprehensive data set, also data analysis becomes more and more tedious. And this is also an aspect that we are working on at the moment. It seems to me that all mass spec vendors, the automatic data integration and data management is fine as long as the peaks are very high. But if you're approaching the LOQ, actually the data, the peak integration becomes less and less reliable, and you really have to do a hand queue of the data. And what I got information from other colleagues, it's like everyone faces this problem. And of course, it would help us a lot if there would be a solution that peak integration becomes more reliable also at lower concentration, because for us it's at the moment the absolute bottleneck.
I understand. I think this message goes out to all the vendors in the world of mass spec. And with that, I would like to finish our session today. Thank you very much, Michael, for providing these valuable insights. And it was really great to have this deep dive analytical session with you today.
Goodbye also from my side.
If you found today's conversation as enlightening as we did and want to stay informed about future episodes, remember to subscribe to our podcast on Spotify or YouTube. By subscribing, you'll never miss a bite of our delicious insights into food safety and analysis. And for those of you with burning questions or lingering curiosities related to our today's episode or any other food safety topic, don't hesitate to contact service at lva.at. Your questions will be addressed either directly in our next episode or by our team of experts on our website, www.lva.at. Thank you for tuning in, and until next time, stay safe and stay informed.